Chief of the Mathematical Biology section at NIDDK. And also he's an adjunct professor in mathematics at University of Pittsburgh. Carson received his Bachelor of Applied Science in Engineering from University of Toronto and his PhD in Physics from MIT 1992. 2013, Carson got the NIH Director's Award for his research excellence. And today he will talk about gene expression, gene transcription from multi and single cell experiments. So please welcome Dr. Carson to the University of Michigan. Thank you so much, Carson.
Thank you very much, Indika, for inviting me here. I've had a really nice time so far. And so I want to tell you about some of the things that I've been interested in and in collaboration with some experimentalists at NIH. And so we all know the central dogma of biology, or molecular biology. So we have DNA transcribed into RNA and translated into protein, and protein then, in the form of transcription factors, reacts back on DNA. And then also more recently, we know that RNA interacts directly with DNA. In fact, maybe the DNA loop might actually be the dominant feedback mechanism. And also we have the environment also affects gene transcription, and I will be focusing on hormones and steroids in particular. So steroids are small molecules, and they're involved in basically all of your body's functions. And so steroids are small molecules that can get inside the cell. They bind to nuclear receptors, which then translocate to the nucleus and then bind to some hormone response element and then initiates transcription. So here's the cartoon version of what goes on. We have steroid, it binds to receptor. The receptor binds, usually there's two, and it's a debate whether they dimerize and bind or bind and dimerize, or are they independent. And then we have our transcription factors, and polymerase 2 then comes along, and we have transcription, and then we go to translation. So I'm interested in this process and trying to understand what exact, can we turn this cartoon into real mechanistic understanding? And so the experimental tools, and I just broadly classified them as multicellular and single cellular, right? So originally the technology, you could take an in vitro assay or living cells or cultured cells, and then you would look at them all up. You can mash them all up, or you could put a reporter into them. You can look at various reporters, right? So we know GROSEQ is a Michigan's big thing. So, and we can look at it over time, right? We could freeze it in time. I'm going to be talking mostly about the steady state, so after it's reached some kind of steady. And we can also look in single cells, and initially what they did was that they would insert multiple genes of the same type, so these tandem arrays, because the reporters and the optics weren't so good initially, and so you needed a lot of them expressing it. And so then we could image a single cell, and you could see lots of the same gene expressing in a single cell. But, and this could then actually give you the actual time course, and you could start to see the differences between cells as they express genes. But then it would still be combinations of genes in a single cell. So the technology got better. Oh, yeah, and you could also look at this in various ways, and you could actually have a reporter onto mRNA, right? So mRNA does not turn into a luminescent protein, but what you can do is you could put in stem loops into the mRNA, and then you can design a probe, some bacteriophage probe or some other type of probe that then attaches to these stem loops. So you can actually image the mRNA itself in live time. And in fact, you can now do it in single cells, and you can actually do single molecules. So you can image individual mRNA molecules. So the technology now is such that you can insert your stem loop probe into the gene. So as the mRNA comes out, you can stick in, and you can even choose whether to put in introns or exons or introns. You can put your stem loops in, and you have this probe that's, you have another gene that's endogenously expressing this probe, bacteriophage coat protein, which will bind to these stem loops, and so you can actually see transcription and splicing even in live action. And so my colleague Dan Larson is like doing this in MIT, and so now we can actually see individual genes, and the goal next is to do multiple genes in the same cell. And eventually we want to do multiple pairs of genes, various parts of the chromosome, and to image that. So that's the state of the art, and the question then is, what sort of insights can you draw from it? So I'm going to just talk to you about a small, my kind of biased worldview of this piece, but there's lots to be done, obviously. And of course, you can look at the protein level or the mRNA level. So I'll be talking about both. I mean, most of my data will be looking at the protein level, but there is evidence that the mRNA at least is indicative of the dynamics of the protein, or at the steady state levels of the protein, if not the dynamics, or if not the abundance. So the main experiment I'll talk about is kind of an old-fashioned experiment with my, I can say about my collaborator, Stoney, who's probably one of the premier biochemists of his generation. So what Stoney is interested in is in steroid-regulated gene expression. And so his prep, he transfects a reporter plasmid that has a GRE in it and a luciferase reporter on it, transfects it in, and then he transfects steroid receptors, transfects other various cofactors, or adds molecules depending on what they are, adds steroid to the thing. And the question then is, you add steroid, you measure photons. And you want the connection between the amount of steroid you put in and the amount of photons that come out, right? So that's, and if you measure the dose response curve, and so Stoney does these very, very careful labs, does multiple replicates and various doses. So you add steroid, various doses, and then you measure the luciferase luminance coming out, right? So literally it's left, the y-axis is photon, and the right is. And if you look at the curves, it fits this Michaelis-Menten function. Okay, so what did I do? Do not touch that. What's the laser? That's the laser. Okay, so sorry. So this function has many names, but it's basically just this function, the Michaelis-Menten function. It's non-cooperative. It's non-cooperative because the Hill coefficient is 1, and it's only got 2 parameters, right? There's the EC50, is kind of the potency of it, and the maximal activity. So if you do this in his prep, and now it's been validated in other nuclear receptors, and probably other genes, although I don't know for sure, but at least for some different types of nuclear receptors, you get very, very good fit. Okay, so this is not an accident. This is really, signal. And so the question is, why? Why would you have Michaelis-Menten? Some people don't think it's, special at all. But for me, when I first saw this data, my immediate thing was, oh, why would you have that? And why is it not, S to the 1.7 or something, right? If you randomly fit data, you hardly ever get 1 as your code coefficient. So, but to me, that was interesting. It was funny that, I was at this retreat at NIH, at NIDDK. Stoney gave his talk, and I was immediately thinking, that's an interesting mathematical question. Stoney walks up to me, because I talked right before him, and I talked about something, I forget. And he said to me, he said, this is an interesting mathematical question, so we started that collaboration right there, which is how almost all my collaborations kind of start. So the classical explanation for why this is true is that steroid has to bind to receptor, right? So if you remember from your first biochemistry class, I was a physicist, and one thing I had learned as a physicist is that physicists know no chemistry, they know no biology, and they absolutely know nothing about statistics. They don't even know what the field of statistics does, is what I realized as a physicist. So as I was in a math department for 5 years, pretending to be a mathematician, to the point that I actually believe I'm a mathematician now. And then I was at NIH, and at NIH I had to learn that there's this field called statistics, there's this field called biology, and there's this field called chemistry. But everything I've learned on the street in some sense. And so the classic steroid receptor, the Michaelis-Menten equation is actually about 100 years old. And it's also an interesting, I tell these sociological stories just for the hell of it. But Menten was a woman, Maud Menten, and she was a Canadian woman who got one of the first MDs in Canada, and she couldn't get a job in Canada or US. But Michaelis was a German chemist, and he decided to take her as a postdoc. So she went there, and they did that first famous experiment. And then she was a professor at the University of Pittsburgh, and they made her a full professor the day before she retired. So, I mean, that's kind of 50 years ago. So I kind of, we're still not leaning in all the way, but we're leaning in more than we were 50 years ago. So, so how would you model this? Well, so if you assume the mass action limit, then that is just simply the reactions are proportional to the amount of stuff, right? More stuff, faster reactions, and then there's these rate constants forwards and backwards. You can write differential equations governing the concentrations, and then at equilibrium, okay, so we're interested in equilibrium because in Stoney's experiments, he adds steroids, he waits 3 days, he measures photons. So you can safely say they're in equilibrium. It's a sealed jar, nothing's going on. You set that equal to 0, then that you can solve that, and you get that the amount of the complex is just the product of the two constituents, and then there's this coefficient of proportionality, which is 1 over the dissociation constant. Or the inverse of that would be the association constant or the equilibrium constant. I like to call it the affinity constant. And then, and the goal is we want to derive RS as a function of S. Okay, so we know at least it's proportional to these two things, but we need to eliminate the other factors. So the second condition is that stuff is conserved, right? There's the mass conservation. So R plus RS is a constant, and so the amount of receptor is conserved and the amount of steroid is conserved. But generally, you're in the regime where you have an abundance of steroid. And so you can just assume that the free steroid concentration is the total steroid concentration. Because the amount bound is very little. And if you make that assumption, then you can just plug in that original equation, RS is equal to R times S, into that equation, solve for S, plug it back, and you get the Michaelis-Menten. Okay, this is actually half of the Michaelis-Menten equation. Michaelis-Menten equation is an enzyme equation. So this would then feed into the next level, and then you would get the velocity. But I'm in equilibrium, so it's actually easy Michaelis-Menten, right? Michaelis-Menten is actually 1 step more difficult. And so mathematically, this is an interesting equation because it's a bilinear equation. Right? So it's linear in R and it's linear in S individually, and the nonlinearity is just the multiplication of R. And this will be a very important algebraic fact, which I will exploit for the rest of the talk. So the Amax depends on the number of amount of receptor, and the EC50 just depends on the affinity constant. Right? So the argument has always been that we have Michaelis-Menten, and the EC50 is just the binding strength of steroids. So if you just simply change the binding strength, you change the EC50. Right? So that's the argument. But then Stoney started to add stuff, and he said, well, what if you add stuff? So this stuff is UBC9, which, I have to say I really don't know what UBC9 is. I think it's got ubiquitin conjugating something. But, it makes Amax move and it makes EC50 move. Okay. So and it's still Michaelis-Menten. So it's still Michaelis-Menten, these things move. So given that they move, then this can't be right. So then the question is, you still have Michaelis-Menten, and it's not because steroid receptor binding is the rate limiting step. So what's going on, right? So that's why it really becomes an interesting biochemical mathematical question. So remember, we have our model of steroids, right? And these are basically from chip-seq kind of experiments, or chip-chip, really, or chip, really. And it's just that we know that these various transcription factors bind, and we know where they bind. And so they make these giant, we think that they make these giant complexes that sit on DNA. And then we know polymerase binds, and then we know there's transcription, right? So we know that classically. And so the simplest kinetic scheme for this, and so this is all biochemistry at this point, is you have something, steroid, binds to receptor, it makes a complex. And then this complex, binds to something else, and it makes another complex, and it makes a bigger and bigger complex, right? So that's kind of incontrovertible to suggest that at least a minimum model of gene expression is the building of complexes, because then you finally attract polymerase, and then you go. And so I'm going to write this abstractly and just call the y variables products, the x variables, I'm going to call them accelerators. And there's a long history of why they're called accelerators. If you know enzyme kinetics at all, any biochemist, they're really just activators. But activator was used already as a term. And so to not confuse it with co-activators, we had to invent a new term. And whenever committees decide on a term, you always get the worst possible term. So that theorem has held true. So they're called accelerators, even though I hate that term, and even though I chose it. So the dose response is then the concentration of yn as a function of y0, right? So we want to know, simply that's just something, we solve the system, is yn a Michaelis-Menten function of y0? That's the first question to ask. So we can write down the mass action equations again. And they break down into, this is a reversible system that obeys detailed balance, so we know exactly what the equilibrium conditions are. And then we know that mass is conserved, so we know what the conservation equations are, right? And you can construct this ab initio, and then the three parameters are the affinity constants and the total concentrations of the accelerators. And you know that because things are conserved, so x1 is in y1, and y1 goes to x2, goes to y2. So any x1 has got to be conserved in all of these things, right? So that's why you get this beautiful triangular shape. So you have the system of equations. You have, 2n equations and 2n plus 1 equations, 2n plus 1 unknowns and 2n plus equations. Okay. You combine it together, you get a 2 to the n minus 1 degree polynomial, and you need to solve that to get y0. Okay, so we know from algebra that when n is greater than 4, you can't solve this in closed form in terms of radicals. But we can then, try to get an idea of the shape. So, so what exactly are we fitting, right? So you can always argue, people will always argue, well, like any old function can fit to Michaelis-Menten, right? In fact, there's the Weierstrass theorem, or the Stone-Weierstrass theorem that says on any finite domain, a continuous function can be fit by a polynomial. Okay, so that's a theorem, arbitrarily well. There is a polynomial out there that can fit it. So it may not be so surprising that you could fit it, right? So we need to say, well, can you accidentally be Michaelis-Menten? Does the solution look like Michaelis-Menten? And so one thing that's special about this thing is that when n equals 1, if you look, the difference between 10% and 90% activation is 81-fold. Okay, so that's an invariant. That's kind of a shape invariant. Okay, that's kind of like the exponent of this thing. So it's kind of a surrogate for the exponent. Would be the 81-fold difference between 10 and 90. So then you can say, okay, let's solve this system for just 2 equations, or let's just calculate. Let's just prove to you that this is true. So we have y1 is equal to a Michaelis-Menten function of y0. I can invert this thing and get y0 as a function of y1. And then I can plug in p times the maximum, and I can make p 10% and 90% and divide, and I get this function. And so when I divide y0 of 1 minus p of y0 of p, then I get 1 minus p over p. If I stick p equals 0.1, I get 9 divided by 1, 81, right? So that's why it's 81. So you can see right away that if I had 2 or something, if n equals 2, I get 9. And then the more cooperative you are, the smaller it is, right? The smaller the fold change. And if you're anti-cooperative, it's bigger than 81. So 81 is non-cooperative, smaller is cooperative, bigger. So if you have two reactions, y0 binds to x1 makes y1, y1 binds to x2 makes y2, and you have to solve 4 equations and 5 unknowns. And so then you can then show that, by that y2, you can get y2 as a function of x1. You use the implicit function theorem. You can show that you can solve this thing. And then, and what you end up with is you end up with this quadratic function in y2. Okay? Then you can prove that the fold change is always greater than 81, or greater than or equal to 81. Okay? So that generically, it's bigger than 81. It's anti-cooperative. And in fact, you can sort of see why it would be anti-cooperative, because cooperativity means you need lots of things to make something happen. Well, in this case, you kind of need less than things, right? Because things get lost along the way. So you're less potent than you would be if you were just one reaction. If you're two reactions, you're lost in the middle. And so you're going to be, so the more reactions you have, you're going to be anti-cooperative. Okay? So this is a way of generating anti-cooperativity. And the reason it's not cooperative, the reason that it's not Michaelis-Menten is because it's not bilinear. Okay? And so this thing goes the wrong shape. So if we were to throw away this quadratic term, then you would see that y2 is equal to a Michaelis-Menten function of y1, right? So we need to throw away this nonlinear term. So how do you throw away that nonlinear term? Well, if you were to just simply say that x1 is not conserved in y2, that really was only conserved in y1, and what you do is you end up with two different Michaelis-Menten functions independently, and then if you stick one Michaelis-Menten function into another Michaelis-Menten function, you get a Michaelis-Menten function back. Okay, that's a nice property of Michaelis-Menten. Right? Is this just an accident? Well, not really, because we know from 100 years ago that the linear fractional transform, or sometimes called the Mobius transform, or the photographic group, forms a group under function composition. So meaning that if you plug the function into the function, you get back the same function shape. It's closed under composition. But if you put any other function in there, you don't get that, right? If you had z to the 2, you plug in z to the 2 into z to the 2, you get z to the 4th, and then you get z to, right? You don't, if you get z to the 1/2, you plug it in, you get z to the 1/4, it'll shrink, right? So 1 is the critical point in function composition. It's an unstable critical point. And it has a representation. It's homomorphic to 2x2 invertible matrices. So you can convert functions substitution into matrix multiplication. And in fact, Michaelis-Menten is even simpler than the Mobius transformation, because that's a complex group. You can take a real version of the group. Really what you can do is you can convert them into lower triangular matrices. So if you just write these functions down as matrices, and you multiply the matrices, and you convert that back into function, you can compose two reactions. Right? So when you see Michaelis-Menten in biochemistry, nature has discovered group theory. It's discovered, an operation that's invariant. It's found a set of objects that's invariant under that. And there's some strong biological reasons why you want that, which I'll get into later on. So that's one way you can preserve Michaelis-Menten, is if you have Michaelis-Menten functions at each step, and you compose them. The second way to compose fractions, and this is very relevant in my household because my third grader is learning fractions right now. And she's learning, sort of learning about common denominator, sort of. So, if you have two fractions with the same denominator, then you add them together, you get the same denominator, right? So you can dress this up and say that this is a vector space, right? You have our vectors, and you can multiply them by constants, you add them together, and you get. So Michaelis-Menten is preserved by two algebraic structures. It's preserved by the group of lower triangular matrices, and it's preserved by the vector space of addition. And we will. So then, using this knowledge, we can then say, what is the most general form of the conservation equation that respects these two groups? Okay, and so this is it. I don't have a theorem that this is it. So at this point, it's sufficient. I don't know if it's necessary. I don't know if it's only if. Any algebraists in the audience want to prove it, that'd be great. Please talk to me. I always need, you can't run out of algebraists as far as I'm concerned. I know Michigan's actually a center for algebra too. There's a lot of excellent representation theorists too. We actually stole one of your guys, Tom Hales, to Pittsburgh. I personally stole Tom Hales because he was my buddy in grad school, and I said, hey, Tom, why don't you come out? We could play tennis. And so he came to Pittsburgh, and then I left the year after he came. Because my wife, who was in Baltimore, stole me from, anyway, that's complex. So you lost Tom because of me, so I'm sorry. So the general form, this is the general form, and there's this one weird step in the middle, which I call the concentration limiting step, and for reasons. So what it is you get pairwise conservation before the CLS, and then you just get superabundance of factors after it, and then at the concentration limiting step, you have everybody. Okay, so this is the most general form. And then everybody afterwards, you can add together to go to product. So if you have this form of conservation, you're guaranteed Michaelis-Menten. And also the Michaelis-Menten is exactly solvable. Okay, you can write down a closed form. So you don't need a CPU. In fact, you could do it, on pen and paper. You can write down the form. So what are the applicable? How does this relate to biology? So what this says is that this is the general form for the binding thing, and what you need to do is you need downstream concentrations to be weak. So what you need is you need weak binding, or you need transient binding. You need the lifetime for these complexes to be very short-lived. Okay, so that's one prediction of this model. And so what you need is you need these products to be small, and so that just implies that the affinities are small. So before the CLS, accelerators are limited. And then after the CLS, you have free accelerator is equal to accelerator. And so that means that you have superabundance, or you have limited products or superabundant accelerators. So, the pithy way to think about it is that you have one region where you have very, very limited transcription factors or accelerators, and then afterwards you have too many. And then you have a transition region in between, which is kind of like the rate limiting step. It's when concentrations are limited. That's why I call it the concentration limited step, although technically the concentrations are limiting here. And so this is where they all kind of bunch up. So this is not the limiting step, it's the limited step. And that grammatical difference makes all the world to me and means nothing to anyone else on the planet. So the prediction is downstream products are weakly bound. So it's really a post-diction because this experiment was done before my prediction. But in the realm of registering your prediction before and multiple comparisons and all that stuff, I didn't know about this. So here's a very famous by Jim McNally and Gordon Hager 15 years ago. What they did is they tagged nuclear receptors, steroid receptors, And so here they are floating around in the cytosol. And and then this really bright spot is where they're bound to DNA. Okay, so they're floating around. Some of them are bound to DNA. Then they took this spot and they shone a bright laser on it and they bleached this area, right? So now they're dark. And so what happens is that if you see the dark spot go bright again, it meant that the dark guy went away and a bright guy replaced it, right? Because the lifetime of going bright is infinity, right? Once it's dark, it stays dark, or longer than this experiment. And so what happened is that within seconds of bleaching, the spot is bright. Okay? So prior to this experiment, people believed that steroids bind to receptor, steroid binds to DNA, and it stays there for the day it takes to transcribe the gene. Okay, that was the old view. McNally comes along and says they're bound for 6.6 seconds on the good day, maybe 2 seconds. And everyone said, there's something wrong with your experiment. So they did it again and again and again. And now I think people believe this. Hold on. Yeah. Gordon is still extremely bitter about this whole thing. But so that confirms the experiment. I mean, that when we showed this to him, I did this thing and I knew nothing about steroids or anything. I knew nothing about genes or anything. And Tony said, hey, why don't we show this to Gordon? He'll be excited. I didn't know Gordon, I didn't know who he was. I'm like, okay, sure. He was like all excited. We told him, and then I realized later that, oh, it's Gordon Hager. It's like, I'm an idiot. And, but we predicted his experiment, post-predicted his experiment, so he's happy with that. So, another thing is that we know that transcription burns ATP, right? So it's irreversible, and what I modeled it with was a reversible process, right? So, but you can replace everything I said with an irreversible process, right? So you can have the blue ball hits the red ball, and it turns the red ball green, which then goes back to red. Okay? So Victoria is a neuroscientist. This is an excitable system, right? And an excitable system is a classic it's a non-reversible dissipative system. And so, so what you could have is you could have X1 bumping into Y1, turning Y1 into Y2 and running away. Okay? So that's just like his 6 seconds. So it left a mark that did something. And then X2 then bumps into Y2 and turns into Y3, and then Y3 goes back. Okay, so you can have this. And then you could have something happen differently, is that instead of X1 bumping into Y, you could have Y now bumping into Z and turning Z into something else. And that's the concentration limited step. Okay? And so later on, I'll show you that the concentration limited step is always the DNA, which kind of makes sense, is that you have steroid binding receptor, other factors start to modify and activate the steroid receptor complex. Then it binds to DNA, which is passing the baton to the next template, and then stuff binds to DNA. And so that seems to be consistent with what we observe. And so that would be another biophysical interpretation of what's going on. If you have these fast events that leave marks. And then you have this change of temperature. So what else do people know? So then I showed this to Dan. Then Dan Larson came, and he came around 2010, and he was one of the first Stedman investigators in NIH. We have this big search every year where we do all our hiring on one day, basically. We have this big festival where everybody comes and gets a job interview on one day, and then we pick the best. It actually happens on multiple days, but pretty much that's how it works. And so Dan was one of the first selected out of 1,000 people who applied. He was one of the 8 that was hired, and he gave his job talk, and I said, I have a theory for you, and he's like, who is this guy? But we're now like good friends and collaborators, which shows that persistence always pays off. So what Dan found was that if you were to image single genes, what you get is you get a Poisson process, effectively. And that you can model this by what's called the random telegraph model, which is essentially inactive goes to active, active goes to transcription, and that's it, 2 steps, okay? And so it's basically one rate limiting step, right? So the nice thing about group theory is that what group theory is all about is that lots of matrices you can marginalize over nuisance variables, right? And so that's just saying I can replace all my reactions by one reaction. And this is just a simple A to I reaction, effectively. And so that has exactly the same statistics as you would see from the random telegraph model. Right? So when you have this magical property of these sequences with these fast weakly things, you also magically get a Poisson statistic as predicted. This time it was a prediction because I had the theory before Dan had the experiment. So I get to count that as a check mark. So it matches, even though it's a biochemical model, you know, maybe fabricated in the imagination of a crazy person, it actually can match some real data. But originally, why do we have this theory? Okay, it's not just to prove that math is useful in life. We wanted to predict mechanisms, right? So we know that UBC9 made Amax go up and made EC50 go left. And so how can you do that? So, I mean, one of the things that we really did was we really showed that gene transcription could just simply use the biochemistry of enzyme kinetics. Okay? That whatever is known in enzyme kinetics can just be transported over to gene transcription. It's just that instead of rates, you go to equilibrium. So it's simplified enzyme kinetics. And in enzyme kinetics, you have inhibition, right? So if you have this reaction, you can have a competitive inhibitor, which just eats the accelerator. So it competes with the accelerator. You can have an uncompetitive inhibitor, and that thing eats the product. And then you can have both on at the same time. It's called non-competitive. Okay? You can have all these inhibitors, but because we use the word accelerator, the opposite of accelerator is decelerator, we call the inhibitor decelerator. Okay? So I'm sorry, I inflicted bad terminology on the world. Instead of calling them activators and inhibitors, which is what they should be called, they're now called accelerators and decelerators. And then you can have, when the decelerators act, they make another thing, and that new product can also go to product, and that's called a partial inhibitor. You can have linear inhibitors, partial inhibitors, competitive, and so forth. You can incorporate all this into the sequence, and you still get Michaelis-Menten. Okay, so you still get Michaelis-Menten. And you can solve it exactly, right? So we have that every step can be modeled as a Michaelis-Menten function, so it can be mapped to this matrix. And that all the pre-CLS steps look the same, all the post-CLS steps look the same, all the CLS steps look the same. I'm okay for time, right? And then you can write down exactly what these matrix coefficients look like in terms of all the parameters in the model. Then you multiply the matrix together, and you have a formula. Okay? You literally can write down in closed form the dose response curve for n reactions where n can go to infinity. And it's in terms of 4n, I think I calculated 4n plus c unknown parameters. Okay? So you can say, well, big deal, you know, anybody can overfit a model, right? How can this be useful? Well, it can be useful. So we have this formula, right? It can be useful because, again, because of the group property. But because of the group property, remember, we can telescope. So even though there's lots of reactions we don't know about, and there may be, you know, an uncountable infinite number of parameters, they can always collapse down to 2 parameters. So there are these 2 effective parameters that characterize all of them. And there's 2 parameters here, and so you can always reduce it to a number of effective unknown parameters that are combinations of the other parameters, right? And mathematically, what that's speaking is that even though you have lots of parameters, they live on a low-dimensional manifold, and so you have to parameterize the manifold. And the manifolds are all you need to know. And so then all you need to do is you've mapped it to topology, because the equations have different topological structures depending on where they are And if you can match the topology of the data to the topology of the model, then you can make a prediction without actually having a quantitative knowledge of the parameter. Okay? So you only need to keep as many reactions as you need to still use this thing. So if you have this dose-response curve, even though it's written in terms of a ridiculous number of parameters, if you only cared about 2 accelerators, then it looks like this. Then you just have to fit, 5 parameters. And if it's an accelerator and a decelerator, it looks like this. And you can do all the cases, and there's only a finite number of cases. Okay? Just a number of combinations of how many 2 things can be, one in front of the other, one in wherever you are. So you can write formulas for all of them, and then you can fit it to the data. And if you do this, you fit it to the data, you can fit this 3-dimensional data. You vary UBC9, you vary GR, which is the receptor. You can make a fit, and then you see that the best fitting model is that UBC9 is an activator accelerator that acts after the CLS. So the prediction is that whatever the CLS is, and I'll show you later data, the CLS is probably DNA. It's acting after GR is bound to DNA. So that's the prediction. And you can do this for all the things you want. So Stoney, who was trained as a biochemist, said, well, in enzyme kinetics, nobody fits to the actual thing. There's this thing called Lineweaver-Burke plots, and we just plot graphs and we make predictions. And I'm like, oh, really? You plot graphs? Okay, so I read these papers, and I'm like, oh, yeah. So what we can do is we have this function here, right? But the function is really in terms of 2 parameters, and these parameters are then in terms of other parameters. So we can just fit the parameters instead of the function. And the nice thing about the fractional linear transform is that once you have fractional linear and you're stuck in the group, anything you look at is always fractional linear. If you take a parameter of something, it's always fractional linear. If the function is fractional linear, it's a fractal of fractional linear, right? No matter what you look at, you can only end up with linear or fractional linear. So if you look at Amax over EC50 and 1 over EC50, these turn out to be the salient parameters. Prior to that, people always just looked at Amax and EC50, but I told Stoney, I said, no, you got to plot Amax over EC50 and 1 over EC50. And if you plot them, they're always going to be these fractional linear functions, And if you're at the CLS, then what you predict is that as you change the amount of that accelerator, if it's a CLS accelerator, then it's literally going to be linear. Okay? Linear in the mathematician sense. Linear. The 1 over EC50 will not change. Okay, so if the potency doesn't change and the Amax over EC50 is linear, then it's an accelerator CLS. If you invert Amax over EC50, if you look at EC50 over Amax, and you plot that thing as a function of the cofactor you're adding, and you get an affine function, right? So which is a linear function with a y-intercept, then that predicts it as a competitor. Okay, so you can just start to say, I just graph these things, and whatever they look like, I can make predictions. And I can predict where they act and what they are. Okay, and so if I have an affine function for an accelerator, then I have, and the 1 over EC50 is affine linear, then that's an accelerator. So here's reporter. Okay, so in Stoney's case, his reporter is GRETK Luke. It's just a plasmid with a luciferase protein coding, DNA coding for a luciferase protein with a GRE element attached to the front. And then he sticks this into the genes, and now we just change the amount of the reporter, and you find that you get this linear curve, and it goes, exactly through the organ. And so we know that reporter is, and the 1 over EC50 does not change. So the reporter is CLS, and it turns out in every system we check, reporter is always CLS. So it was not, it wasn't a fluke. We can look at these other things. So Stoney throws things in called S-Smart and TIF2. I really have no idea what these things are, but they change the curves. And so, in fact, Stoney and I play a game so we don't bias the predictions. He just says, here are some graphs. Tell me what they are, and I don't know what they are. And so I make a prediction for him, and he's like, oh, that's because of the, this experiment from 1987 or something. So it was a great collaboration because I didn't have to know anything and I could still contribute. So we have, so the prediction then is, oh, it's an affine function, EC50 over max. So we predicted that S-smart is a competitive decelerator at the CLS, and TIP2 is an accelerator. So we can start to classify these things and say which one acts before what. And at this point, they may not have any meaning, but at some point, my science fiction fantasy of this is that once we start to know what these things do, we can start to design drugs to actually do these things, right? Say you want to upregulate a gene and you only want to upregulate a gene in the pancreas, but not in the liver, and we can try to design a drug that changes, maybe this is the cofactor we can change, and you'll start to actually titrate the amount of genes, okay? So I'm thinking that the next generation of drugs, right, we've run out of GPCRs to target. We've run out of nuclear receptors. We don't really know what else to target. Well, the next thing to target is to actually target gene transcription. And we know these are the things where we can, so we need a mechanism. That's why, biochemistry and biophysics is still useful, because we need to know what we need to do, right? So I know that this is, Michigan's the center of bioinformatics, and you can tell us which genes are important, but then you need to know how do we change the genes, right? Like I was saying at lunch, do we just CRISPR everything? Is that it? Is that the ultimate therapy? We throw in a CRISPR for every cell in your body and we just swap every single, I don't know, maybe that is what's going to happen. And this is all useless. But, prior to that technology, maybe we can do this. Or at least, pharma might want this more, Anyway, so I kind of run out of time, right? Or I got 5 minutes. Okay, so that's, there's also steroid-regulated repression. In fact, repression might actually be more important than induction. So interestingly enough, if you add steroid to some genes, it makes it go down, and it's also linear fraction. Okay, so it's not Michaelis-Menten anymore, it's this linear fractional function. And so then the idea simply is, well, maybe all gene expression is linear fractional, and that steroid in one role is the inducing agent, and in the other role, it's just suppressing some other guy who's the inducing agent. Well, I just said, okay, well, then if steroid's acting as a thing that's suppressing activity, what can it be? Well, it could be a, right? So previously had induction, now we have something else. This is AP1, PMA. This thing induces the gene. Now steroid, steroid could just compete with some factor. Steroid can compete with a factor at the CLS. Or CLS could be an accelerator after the CLS. And one of the things we showed is that you could have acceleration after the CLS because it could divert an effective pathway to a less effective pathway, right? So there's 3 ways, qualitative ways you can inhibit. And so then what you can do is you can derive the whole theory again for repression. You can then say, well, what does GR do? And you can then look at all these different plots. And you can predict that all these things, AMAC, AMIN, AMAX, IC50, and then this combination, the prediction of the theory, these things will all be linear fractional functions. You can sort of show that it's true. And then you can then try all the models out and see which one is it. And the prediction is that GR is an accelerator after the CLS. So the difference between gene and steroid-regulated induction and repression is that in induction, it is an accelerator before the CLS, and in the repression, it's an accelerator after the CLS. So in some sense, the mechanism of GR is conserved. It's just where it acts is different between induction and repression. So that's one of the predictions of the model. And finally, there are these partial agonists, right? There's tamoxifen and raloxifene, RU486. And these are all things that are antagonists to steroids, right? So one major drug class we have are antagonists. And so the question has always been, how do antagonists work, right? So people have always thought that an antagonist, it binds to the receptor and occupies the receptor, and that makes the receptor not work anymore. Not work was to be defined. And so you can define partial agonist activity is just simply take the AMAX with the partial agonist to the control. And in this case, control is dexamethasone. And then you can change GR, and you can show that one of the things you find is that if you change GR, you get it. You still get difference. So one of the things Stoney showed was that partial agonists were not always partial, and in fact, if you throw in enough stuff, you could actually turn a partial agonist into a full agonist. So we could then throw the theory at this thing, and we could say, well, what can you say about partial agonists? Well, one thing you can say is that what does partial an agonist do to reduce activity? And so one of the things you can predict right away is that affecting steroid receptor binding affinity cannot affect the amount of gene expressed. And the reason is quite simple, and that is because steroid receptor binding only affects the EC50 of the amount of steroid receptor complex and not the total amount. Okay? So if you add lots of steroid, it doesn't matter what the binding affinity, right? So people had known this experimentally. They didn't really know why the agonist activity binding affinity didn't matter, and this is a simple explanation for why. So what is it doing? Well, what it's doing, the model just predicts it affects the binding affinity of some other step after steroid receptor binding, right? So some antagonist binds to the receptor, it forms a different complex, and this thing then has a different binding affinity, right? So people previously said, oh, well, it breaks it, right? Just doesn't make it bind. But it doesn't have to not make it bind, it just has to make it bind differently. And then you can then just write down the formulas like we did before, and you can write them down. And one of the predictions is that these parameters are in terms of the other parameters of the model, that if you start to increase some receptor, you can always alleviate a partial agonist. And in fact, depending on where the partial agonist is acting, if two different partial agonists have different activity, but they cause the perturbation at the same step, you will show that if you add enough of this factor, you can get them to saturate at the same level. Okay? So the prediction, and in fact, if you can show that if this next step is immediately after steroid receptor binding, C3 and C1 are both 0. Then as x goes to infinity, this thing goes to 1. What that means is that if you add enough receptor, you can turn the partial agonist activity to 1, that they all saturate to 1. So previously people have thought that you had an agonist, it lowered by 30% compared to the control. So that if you changed receptor and you increase control, you would increase agonists and the same ratio would be the same, right? But what this theory is showing is that no, you get convergence. That even though the control goes up, the other one goes up more. And so, this data may or may not be showing that. And so the prediction is that if we could go to enough plasmid or add enough UBC9, these things would all converge. And so these experiments were done like decades before the theory, and Stoney retired, now biking in Romania or something. And so this is the next thing to test would be. So thank you very much. I'll end here. Most of the stuff I presented was I had a summer student who stayed for 2 years, Karen Ong, who's now finishing her PhD at Courant now. And then I've been collaborating with people, Dan and Antoine. We're doing lots of single. I usually put my slides on my blog. I have this blog that I've been keeping, and so versions of this talk already exist. But I probably in a day or two I'll put up the actual slides for this if you're interested in looking. So thank you very much. I didn't go over too much, right?
So now this one works. So, well, it's very interesting, the work. So one thing that I don't fully understand is why you say that it's non-Michaelis-Menten. So in enzyme kinetics, people generally say that you have Michaelis-Menten as long as you have the form of Michaelis-Menten, and then you have this apparent Bmax and apparent Km. That essentially you have mechanisms like inhibition and activation that would shift according to the mechanism.
Yeah.
So why you argue that then it's not Michaelis-Menten? So
no, I didn't say it was not. Okay. I said it is Michaelis-Menten. It is Michaelis. I mean, it's simplified Michaelis. Michaelis-Menten is always talking about the rate, right? Because it's Usually it's an enzymatic thing where you have some flow and you want to predict the rate of the reaction. So that's the literal Michaelis-Menten. I'm just saying that it's the equilibrium part of the Michaelis-Menten. It is exactly the same as Michaelis-Menten.
Okay.
If I said it wasn't, I didn't mean that. I mean, either I made a mistake or I don't know what I said. Thank you. Thank you.
Biology of the prototype receptor, right? I mean,
Yeah.
I don't know if you know Keith Yamamoto's work. Uses
Yeah, absolutely. Yeah.
Repression and stuff.
Yeah.
How that works differently.
Well, I mean, I'm on record, right? But I think that I can explain Keith's data in terms of my framework.
I do too.
Keith may or may not.
He may not.
He doesn't believe that I can explain it.
Yeah, I know.
Yeah, but
he's a problematic.
But I think I can explain his differential expression data exactly in terms of that the steroid acts as a inducer in some places and an inhibitor in other places, and that at baseline it's just simply it's quantitative, not qualitative, and it's not really this incoherent feedback feedforward thing that he thinks it is. I think it's this, but you know, I'm not going to argue with Keith. I mean, if Keith doesn't believe me, it's okay.
But I think you should say. Group theory is found biology. Group
theory is found. Yeah.
You said the other way around.
I'm sorry, I said that.
Yeah. But I mean, it is really something knowing a lot about prototype receptor.
Yeah. No, but I actually think. I mean, one thing I didn't get into. I think that you want to have a group because of the evolvability, because you can swap elements out, right? Yeah. Supposing you want a gene where you don't want to have these cofactors acting, you don't want it to break the system, right? So if you're evolving things where you want to like insert gadgets in and out, then you want to just take chunks out, right? You want to just, delete whole sections of, you want a transposon to come in and just, if it removed it, it's okay, right? Because it's still Michaelis-Menten that you're just going to minorly perturb it. But I think that in some sense, biology and group theory discovered each other. I mean, they're a match made in heaven. I don't know, but
I guess I'm a biologist.
I know, I understand fully that biology always comes first, and math is the queen of sciences after all, right? And whoever's king takes turns, but math is always the queen. All right, thank you.
Thank you so much, Hutchinson.